I'm a new assistant professor in the Department of Biochemistry, and today I'll be telling you about a new study supported by Adipocyte to uncover the mechanisms of obesity-accelerated breast cancer. So obesity, as we just heard, is a known risk factor for about a dozen types of cancer. And about 15 to 20% of all cancer deaths in the US annually are attributed to obesity. We know that 1 in 8 women in the US are affected by breast cancer, and again, obesity increases both the incidence of breast cancer by about 30 to 50%, as well as the severity of breast cancer in postmenopausal women. So our goal is to understand why obesity accelerates breast cancer growth. And we specifically hypothesized that breast cancer cells change the surrounding obese breast fat pad to make it into a more tumor-supportive, more tumor-nurturing environment. And so the purpose of our experiments is to identify the factors that are released by breast cancer cells to change the surrounding fat pad, to discover how these changes promote tumor growth and metastasis, and ultimately to test if blocking these interactions can prevent breast cancer cells from growing. So in other words, we're proposing to interrogate the localized reciprocal paracrine signaling network in obese breast cancer. And to do this, we're proposing 3 different aims. There you go. The first is to generally profile how breast cancer cells remodel the obese mammary white adipose tissue. And then we will focus specifically on 2 cell types in the mammary white adipose tissue, the adipocyte stem cell and the mature lipid-laden cancer-associated adipocyte. So we know that white adipose tissue is a highly dynamic tissue. Both the composition and morphology of cells are known to change with obesity and in the presence of cancer. And so to define these changes, we are isolating mammary white adipose tissue from 4 groups of mice, lean and obese, lean and obese, and control as well as tumor-bearing mice. And we're analyzing this tissue using 2 methods. One is whole mount imaging, and the other one is single-cell RNA-seq. And together, these 2 approaches will inform on changes to the relative abundance of cells, to the morphology of cells, and the gene expression profile, and how these change with obesity and with breast cancer, to then identify candidate paracrine signals that can drive obesity-accelerated breast cancer. We're also specifically focusing on adipocyte stem cells. So breast cancer is known to increase the recruitment of adipocyte stem cells to its microenvironment, and this is further enhanced in obese patients. Mouse models have shown that depletion of adipocyte stem cells in the mammary white adipose tissue actually inhibits tumorigenesis. And similarly, co-injection of breast cancer cells with adipocyte stem cells in xenograft models increases both tumor growth as well as metastasis. And again, this is further enhanced if adipocyte stem cells were isolated from obese patients. So we want to understand what breast cancer cells do to adipocyte stem cells. And to do this, we have a panel of human breast cancer cells that we grow in vitro. We take the conditioned media and then apply it to adipocyte stem cells isolated from lean and obese mice to assay the effect on migration and adipogenesis. And then we analyze the conditioned media using mass spectrometry. Similarly, adipocytes are known to be highly secretory, and cancer-associated adipocytes have been shown to promote the proliferation of immediately adjacent breast cancer cells. And so to understand how adipocytes do this, we are isolating primary mature adipocytes, again from lean and obese mice, grow them in culture, and then we take the conditioned media and apply it to breast cancer cells to assay effects on proliferation, migration, chemotherapy-induced apoptosis, and on organoid formation. And once again, we take the conditioned media and then analyze it by mass spec to identify the factors that mediate this effect. So with that, I'm out of time. But to summarize, we're interested in understanding the reciprocal localized interaction between breast cancer cells and the obese mammary white adipose tissue.